Hello guys, welcome to this lesson. We are going to talk about protein tertiary structures. But before starting, I want to tell you in more detail what we're about to learn. So let's see right now. We will talk about the key aspects of protein tertiary structures. Then we will talk about denaturation and reversibility. Afterwards, we will talk about protein folding and its kinetic pathways. Finally, we will talk about Amphensing's experiment. So, let's get started. Protein tertiary structure refers to the three-dimensional arrangement of amino acid residues within a single protein molecule. It is the final level of protein structure and is crucial for determining the protein's specific function and activity. Tertiary structure arises from interactions between amino acid side chains, the R groups, and includes various folding patterns, motifs, and domains that contribute to the overall shape and stability of the protein. Understanding protein tertiary structure is essential for elucidating protein function, predicting interactions within other molecules, and designing therapeutics. Let's delve deeper into the key aspects of protein tertiary structures. So, one of them is folding. Protein tertiary structure is defined by specific folding patterns adopted by the polypeptide chain. These folding patterns result from interactions between amino acid side chains, including hydrogen bonding, van der Waals forces, hydrophobic interactions, and disulfide bridges. The folding process is driven by the protein's primary sequence, with certain amino acid sequences predisposing the protein to fold into particular motifs or domains. Protein, in fact, have often included recurring structural motifs such as alpha helices, beta sheets, turns, and loops. These motifs represent common folding patterns observed in proteins and contribute to the overall structure and function. Domains are distinct structural units within a protein that possess specific functions or activities. They are often formed by combinations of secondary structure elements, for instance alpha helices and beta sheets, and can fold independently of the rest of the protein. Domains may function autonomously or interact with other domains to perform complex biological functions. Stabilizing interactions are also important. In fact, protein tertiary structure is stabilized by a combination of non-covalent interactions and, in some cases, covalent bonds. Hydrogen bonds form between polar or charged amino acid side chains, contributing to the stability of secondary structures and tertiary folding patterns. Hydrophobic interactions drive the burial of hydrophobic amino acid residues within the protein core, minimizing their exposure to the surrounding aqueous environment. Disulfide bonds form covalent linkages between cysteine residues, providing additional stability to the protein structure, particularly in extracellular or membrane-bound proteins. Now let's talk about another important aspect, denaturation. It is a process in which a protein loses its native three-dimensional structure and, consequently, its biological activity due to external factors such as heat, pH changes, chemicals, or mechanical agitation. This disruption of the protein's structure leads to the unfolding of its polypeptide chain and the loss of this functional chain. Denaturation can occur in various biological contexts, including during cooking, digestion, or exposure to environmental stressors. However, it is important to note that denaturation is not always irreversible. In some cases, in fact, proteins can regain their native structure and function under, of course, appropriate conditions. Let's explore denaturation in more detail and discuss the reversibility of this process. Denaturation can be induced by a variety of factors, including heat, extreme pH, organic solvents, chaotropic agents like urea, heavy metals, and mechanical stress. These factors disrupt the non-covalent interactions like hydrogen bonds, and in some cases covalent bonds that stabilize the native protein structure. There are of course some consequences of denaturation. In fact, denaturation results in the loss of the protein's functional shape and often its biological activity as well. The unfolded or misfolded protein may aggregate, forming insoluble aggregates or amyloid fibrillus, which can be harmful to cells and tissues. Denatured proteins may also lose their solubility, becoming insoluble in water or organic solvents. In many cases, denaturation induced by heat, however, is reversible. When the denatured protein is cooled, it may regain its native conformation and biological activity. This reversibility is due to the fact that the primary sequence of amino acids remains intact, and the disruption of non-covalent interactions is often reversible upon cooling. However, prolonged or extreme heat exposure may lead to irreversible denaturation and protein aggregation. Then we also have the case of chemical denaturation. This is denaturation induced by chemical agents such as urea or 1-iodine hydrochloride, and it is often reversible. These agents disrupt protein structure by interfering with non-covalent interactions, but once removed, the protein can sometimes refold spontaneously into its native conformation. As well, we have pH denaturation. This is denaturation caused by extreme pH conditions, which may or may not be reversible depending on the protein and the pH range involved. Some proteins can refold into their native structure when returned to physiological pH, while others may undergo irreversible structural changes or aggregations. Another fundamental aspect regarding protein tertiary structures is folding. Protein folding is the process by which a linear sequence of amino acids, known as a polypeptide chain, spontaneously adopts its three-dimensional biologically active conformation. This process is essential for proteins to carry out their specific functions within cells and organisms. Protein folding is a highly complex and dynamic process driven by a combination of thermodynamics and kinetic factors. Understanding protein folding is crucial for elucidating the structure-function relationships of proteins, as well as for addressing questions related to protein misfolding and diseases such as Alzheimer's, Parkinson's, and prion diseases. Let's see now some factors influencing protein folding. So, in the case of thermodynamic stability, we can say that proteins tend to adopt folded conformations that represent low energy states, maximizing thermodynamic stability. The folded state of a protein corresponds to the global energy minimum on the folding energy landscape. On the other hand, kinetic pathways in proteins folding refer to the series of intermediate states and transitions that a protein undergoes as it folds from an unfolded or denatured state to its native biologically active conformation. Understanding these pathways provides insights into the mechanisms and energetics of protein folding and is crucial for elucidating how proteins achieve their native structures. Kinetic studies of protein folding involve monitoring the rates and mechanisms of folding processes, typically using experimental techniques, such as NMR, nuclear magnetic resonance spectroscopy, and single molecule techniques as well. Now let's see some key aspects of kinetics pathways in protein foldings. So we have folded and unfolded states, as we have said. Protein folding typically involves the transition between an unfolded, which is the nature state, and a folded, native state. The unfolded state lacks a defined three-dimensional structure, while the folded state corresponds to the protein's biologically active conformation, as we have said. Intermediate states may exist along the folding pathways, representing partially folded conformations with varying degrees of structural organization. Let's see now the transition state theory. So, transition state theory describes the rate-limiting step in a chemical reaction which corresponds to the highest energy point along the reaction coordinate. In protein folding, the transition state represents the high energy state at which the protein undergoes the most significant structural rearrangements as it progresses from the unfolded to the folded state. As well, we need to study folding mechanisms. Protein folding mechanisms describe the sequence of events and structural changes that occur during folding. The two main folding mechanisms observed in proteins are the two-state and multi-state folding models. In the two-state model, the protein folds directly from the unfolded to the folded state without populating detectable intermediate. This model suggests a single rate limiting transition state. In the multi-state model, the folding pathway involves the population of intermediate states, as the image you have on your right, each representing a local energy minimum on the folding landscape. The folding process may proceed through multiple folding pathways and intermediate states before reaching the native conformation. Well, let's analyze folding kinetics. So folding kinetics describes the rates at which proteins fold and the factors that influence these rates, such as temperature, pH, and the presence of cofactors or chaperone proteins. Kinetic experiments such as protein folding and folding kinetics measured by techniques like stopped flow fluorescence spectroscopy or temperature jump methods provide insights into the folding mechanisms and energy landscapes of proteins. Now let's see the significance of kinetic pathways in protein folding. So one of them is predicting folding mechanisms. So kinetic studies help elucidate the mechanisms by which proteins fold and identify the intermediates populated during the folding process. Understanding folding mechanisms is essential for predicting protein folding pathways, identifying potential folding traps or misfolding pathways, and characterizing the energy landscape of proteins. There are some implications for protein misfolding and disease. Aberrant folding kinetics or misfolding pathways can lead to the formation of misfolded protein aggregates associated with various diseases, including neurodegenerative disorders like Alzheimer's and Parkinson's diseases. Kinetic studies of protein misfolding provide insights into the factors contributing to disease pathology and may inform the development of therapeutics strategies. As well, we need to know that engineering proteins with desired properties is another important of kinetic pathways. Knowledge of folding kinetics and pathways, in fact, enables the rational design and engineering of proteins with desired folding properties, stability, and functionality for applications in biotechnology, medicine, and material science. As well, we need to talk about the significance of protein folding. So, as a recap, we can say that protein folding is essential for the biological activity of proteins as the folded structure determines their specific functions and interactions with other molecules. Aberrant protein folding can lead to misfolded or aggregated protein species, which are associated with various diseases, including neurodegenerative disorders, cancer, and autoimmune diseases. Understanding the principles of protein folding is crucial for predicting protein structures, designing therapeutics, and engineering proteins with novel functions. Now let's talk about one case of study regarding tertiary protein structures, which I find quite interesting. It is Amphinson's experiment. It was conducted by biochemist Christian Amphinson in the 1950s and 1960s, and it was groundbreaking in order to provide key insights into the relationship between protein sequence and protein structure. These experiments demonstrated that the native biologically active conformation of a protein is determined primarily by its amino acid sequence rather than by external factors. Amphinson's work laid the foundation for our understanding of protein folding and stability, earning him the Nobel Prize in Chemistry in 1972. So let's see the key experiments and findings. So he made an experiment with ribonuclease. So this is his most famous experiment, and it involves the denaturation and renaturation of ribonuclease, an enzyme that catalyzes the degradation of RNA. Amphinson first denatured ribonuclease by treating it with urea and beta-mercaptoethanol, which disrupts the protein's native structure. Remarkably, when the denatured ribonuclease was allowed to refold in the absence of denaturing agents, it regained its native conformation and enzymatic activity. This experiment demonstrated that the amino acid sequence of ribonuclease contains all the information necessary for the protein to fold into its native structure spontaneously. Let's see interpretations from this experiment. So, Amphinson's experiment provided compelling evidence for the sequence hypothesis, which proposes that a protein's amino acid sequence dictates its native three-dimensional structure. According to this hypothesis, proteins fold into their native conformations by navigating a folding energy landscape, with the native state representing the most thermodynamically stable conformation. Let's see some implications. So, Amphinson's work challenged the prevailing notion at the time that proteins required additional factors, such as chaperone proteins or cellular machinery, to fold correctly. His experiments demonstrated that the information needed for protein folding is encoded in the linear sequence of amino acids, and the folding process is governed by the laws of thermodynamics. These findings revolutionized our understanding of protein folding and provided a conceptual framework for studying protein structure-function relationships. Amphins's experiment paved the way for subsequent research on protein folding, stability, and misfolding, as well as the development of computational methods for predicting protein structure. His work has had a profound impact on fields ranging from biochemistry and molecular biology to biotechnology and drug discovery. So now, let's see the significance of protein tertiary structures. So, protein tertiary structure is critical for determining protein function, as it dictates the protein's ability to interact with other molecules, bind to ligands or substrates, and catalyze biochemical reactions. Changes in protein tertiary structure can lead to alterations in protein function and may underlie the development of diseases such as misfolding diseases like Alzheimer's and Parkinson's. Understanding protein tertiary structure, then, is essential for rational drug design, as drugs often target specific protein structure or binding sites to modulate biological pathways and treat diseases. Well, guys, this is all for this lesson. I hope you liked it, and let's see you in the next one. Bye bye.